All right, welcome to this podcast, everybody. And today I'll be talking to you about transcription. And so, if you recall from the central dogma of molecular biology, we'll see that transcription actually falls after DNA replication. It's the process where the DNA sequence is translated or transcripted, we should say, into messenger RNA. And this is kind of the second step in our dogma where it goes from DNA to messenger RNA and then to proteins, and which will eventually be used for functions after folding. So DNA-dependent RNA synthesis, is called transcription. It's basically where DNA is known as the information storage. Serves as a template. All of our genetic information is stored in the DNA. And a fun fact for all of y'all that didn't watch any of the previous videos is that DNA actually is the same in every single cell, even though every single cell has different expression of the DNA sequence itself is the same. And we'll talk a little bit more about what that means later on. So all RNA is made by transcription, which is the process of transcribing the DNA or making it into RNA. And so messenger RNA is actually a short-lived copy of the DNA. It's basically this kind of intermediary step where we take the RNA sequence from the DNA, we transport it out of the nucleus, and we can use it eventually as an instruction manual to construct our proteins. So genes can be encoded on either strand of the DNA, and we can see that this gene A here goes from 5' to 3', and gene B goes from 5' to 3' as well. We have to remember that transcription does occur from the 5' to 3' direction, and it does not occur from the 3' to 5' direction because the machinery of the RNA polymerase does not add nucleotides on from a 3' to 5' direction. We see that also in our DNA polymerase where we have to replicate DNA in a 5' to 3' direction, and where we add nucleotides in a 5' to 3' direction. Our overall replication fork, however, will still move in the 3' to 5' direction. So we say that RNAs are complementary to the template strand. So our template strand will be this strand right here, and we'll call this strand the coding strand. The top strand is the coding strand, and the bottom strand is the template strand. The template strand is complementary to the coding strand, and so because of that, the RNAs themselves are complementary to the template strand, which means they are identical to the coding strand with the exception of thymine nucleotides becoming uracil. And we can see that right here as well. Genome annotations depict gene coding sequences the same orientation as the coding non-template strand. So that's pretty straightforward. And it says, only one strand is converted into RNA. We call this coding strand a coding strand because, it's very easy for us to, transcribe the messenger RNA or let ourselves know what the sequence of the messenger RNA will be using the coding strand. We don't have to, mentally convert the DNA nucleotides into their complementary base pairs and remember to substitute the thymines with uracils and whatnot. We can just take that coding strand immediately and directly read out the messenger RNA transcript, making sure to substitute our thymines with the uracils. So we see here, always synthesized from 5' to 3'. Always remember that. So we actually use this template strand, and we can synthesize the messenger RNA as we move along in this 5' to 3' direction. So RNA is synthesized from 5' to 3'. The direction of transcription, however, is in this direction, which is 3' to 5'. So we have to, distinguish. I think this was one of the more complicated questions on our exam from last midterm. Is that remember that the direction of transcription and the direction of replication for DNA is from the 3' to 5' direction, and where the machinery moves in the 3' to 5' direction. However, the RNA itself and the DNA itself is synthesized in the 5' to 3' direction, where the mechanism actually lays down the nucleotides in the 5' to 3' direction, because we cannot add on to there's no hydroxyl group on the 5' carbon. However, there is a hydroxyl group on the 3' carbon, and we need to use that OH group because it's a nucleophile. We use that to attach additional nucleotides to the growing strand of nucleotides. So RNA will exit as a single-stranded molecule, which makes messenger RNA a very unstable molecule. It makes it extremely vulnerable to exonucleases. And so here we can see an example of coding DNA with template DNA and RNA as well. And we can see this start codon here is the same. And start codon always will be AUG. And we'll talk about that in further videos regarding transcription. Sorry about that. I think my messages were going off momentarily. Let me mute that for y'all. Sorry about that. All right, so transcription overview, we have 3 steps. We have initiation, elongation, and termination. Initiation is how we initiate the transcription, how the RNA polymerase will bind to the promoter in the DNA and form its closed complex, and eventually begin with the start codon and initiate the process of DNA transcription. Elongation is the process of the transcription after initiation, where we actually get the machinery started and producing RNA nucleotides, forming itself into its messenger RNA transcript. And termination, where we finally release the mRNA transcript from the RNA polymerase and allow the RNA polymerase itself to dissociate from the DNA molecule. So prokaryotic, we'll talk actually about prokaryotic transcription, and we're going to talk about eukaryotic transcription as well. So in prokaryotic RNA polymerase, we have a core enzyme that actually associates with this thing called a sigma factor. And the sigma factor actually helps us kind of determine where in the DNA our RNA polymerase will eventually bind. And so RNA polymerase, itself will bind to the DNA non-specifically, but it itself will be guided to a promoter region, which is a place in the DNA that will facilitate transcription to occur. So to think about, where we can get, how do we get this, whole RNA polymerase in the correct position? Let's think about, these kind of sequences. So in a prokaryotic promoter region, we have a -35 sequence and a -10 sequence, both of which are upstream of the start point over here at the +1 site. And we notice at the +1 site that there is no 0 site because the +1 actually marks the start of our RNA transcript. And so, our sigma enzyme, sigma factor will actually recognize these 2 sites and will bind to these 2 sites, the -10 sequence. In reality, the whole molecule will fit on top of this whole region right here. And so this is our template strand, our coding strand, and our nascent RNA chain, which is just beginning to start forming with our triphosphate right here. So chemical mechanism of RNA sequences. It's kind of the same mechanism as DNA sequences. We have a growing RNA strand and recognize that we do need our magnesium factors, our metal ions. You have the incoming nucleotide that will be able to attach to the previous nucleotide through the 3' hydroxyl group, nucleophilic attack. It's pretty much the same as our DNA replication and synthesis. And so we see incoming RNTP, we have all these phosphate groups that this phosphate group will actually detach from the other 2, the diphosphate, so the alpha phosphate group is getting attacked by our 3' hydroxyl, and that itself will then form a bond between these 2 O's, and it will link up and form our growing RNA strand. So sigma factor as we mentioned before, guides RNA polymerase binding to specific sequences, and it also actually facilitates the unwinding of the DNA double strand. Recall that in DNA replication, that enzyme was called helicase, where that facilitated the unwinding of our DNA strand. However, in RNA transcription, our sigma factor can also play that role as well. And so we have specific binding of our sigma to our -35 and -10 promoter sequences. And after it binds to it, it forms this thing called a closed promoter complex. We see that the sigma factor here binds to -10 and -35, excuse me. And then the sigma factor will begin the unwinding of DNA around the initiation site so that the initiation of transcription can occur. Notice that the -35 and -10 are both upstream of the start transcription site. And notice that even though in the simulation didn't show this, but that the sigma factor itself will actually cover the entire region from the -10 to the -35. It doesn't bind to just one, it binds to both because it takes up this whole region here. So unwinds the DNA at the promoter region to initiate transcription. And the second step, transcription, occurs. And notice that no energy in the form of ATP is actually needed for the unwinding because in our prokaryotes, the sigma factor unwinding is actually energetically favorable. And so we have sigma factor 2, which actually binds to -10, and sigma factor, the sigma region 4, excuse me, binds to the -35. And we also have this up UP segment. It's recognized by the sigma CTD of the polymerase alpha subunit. This is just an extra part of our sigma factor that will bind to this UP element, and these just make sure that we are binding to the right regions to allow the initiation of transcription. And this is just kind of these strong promoter regions here in our E. coli. This is we took this off the internet, and we were able to see these are kind of the sigma factor binding sites. We also look at these things as consensus sequences because our consensus sequence can actually tell us how efficient the transcription might be for a certain gene or whatever the mRNA will encode for. A consensus sequence is actually determined predetermined, and how closely, for example, I guess a specific strain follows the consensus sequence will actually determine how well that strain will be transcripted into messenger RNA. And so these are the consensus sequences for our sigma 70 promoters, right here and right here. And so whatever the DNA is, well, wherever we want to transcribe the DNA, if the DNA sequence has these consensus sequences in them, and if they match perfectly, then we know we'll have a high level of transcription. The lac promoter, however, it's an example of a weak promoter because we can see that there's a lot of differences from the consensus sequence. And that means the transcription will not proceed as efficiently as it can. So transcription initiation, we have our open complex formation after the closed complex formation after it unwinds the DNA, and the sigma factor binds to the specific sigma sites, the -35 and -10 sequences. And as initiation continues, RNA binds more tightly to the promoter at the -10 region, and it will be more untwisting of the DNA at the region. And here, the RNA polymerase is orientated, and it will start transcription at the +1 site. After 8 to 9 nucleotides of RNA synthesis, our sigma factor is released because the nucleotides, the RNA polymerase, will actually outcompete it. and so the sigma factor released, and then the process of elongation will occur. So enzyme untwists DNA ahead of it, keeps single-stranded transcription bubble, about 25 base pairs, and remaining exiting the enzyme in a single-stranded form. And so eventually the RNA polymerase will be responsible for the unwinding of the DNA helicase. So let's talk about the steps in our prokaryotic transcription. Initiation, you have formation of the closed complex, then the unwinding of DNA to yield the open complex by the sigma factor. You have the synthesis of 5 to 10 phosphodiester bonds equating to 5 to 10 nucleotides. Release of sigma factor occurs because the RNA begins to form and it outcompetes the sigma factor. And so then we can proceed into our elongation of, for RNA synthesis. Elongation, basically as each nucleotide is added in, base pairing occurs and the backbone phosphodiester bond forms. This occurs at about 50 nucleotides a second, and it's kind of the same process in which DNA synthesis occurs. Very simple. Transcription termination, we have two forms at which termination can occur in prokaryotes. One is our row-independent transcription, and where we actually insert something called a hairpin loop, followed by a run of U's, and also known as row-dependent, which is a protein factor. And these are polysites that become termination sites when we have the presence of this protein factor. So let's talk about row-independent termination first. The RNA polymerase actually pauses, and we have this symmetrical sequence here, which are actually complementary to each other. And this is there because now we can use this sequence and we can form actually a hairpin loop. So it pauses, and the invert repeats in the RNA fold into a hairpin loop, which causes the RNA polymerase to pause. Because this hairpin loop, the RNA polymerase is like, what the hell is going on? Like, we really can't get past this abnormal structure in the messenger RNA. And so then obviously this tail actually has it's a poly U tail, because the poly U tail actually, if you recall, a U and the adenine interaction is a lot less strong than a G and C interaction, because in U and A and T and A, both of those base pairs only are able to bind each other via 2 hydrogen bonds, while with G and C you have 3 hydrogen bonds, and so the hydrogen bonds in this base pair will break, allowing for the transcript to separate from the template, and that terminates transcription. Now, this obviously something we should look for in terms of structure. We want to look at a dyad symmetry, and where in our strand here we see that we have this sequence right here, another nonsense sequence or whatever sequence you may have, along with a perfectly complementary sequence to the sequence that we just showed right here. So you can see that the C will bind to the G will bind to the C will bind to G, and so on. And that actually will create our hairpin loop. And after it's transcribed to the RNA, it will create our hairpin loop, which will allow us to pause the RNA polymerase and cause a dissociation of the RNA transcript from the DNA, from the poly U tail. In row-dependent termination, a DNA sequence actually causes the polymerase to stall. The row catches up, and the helicase activity of the row causes dissociation. Basically, we have the sequence in the DNA. It causes, and this is the rut site right here. And basically, it causes the the RNA polymerase to stop the transcription. And then you have this helicase right here that actually will go in and rip everything apart basically, and cause the dissociation of the messenger RNA. And notice here we have ATP, which means rho-dependent termination is actually ATP dependent. And this is kind of our transcription overview. Initiation with the binding of the sigma factors to the -35 and -10 consensus sequences, remembering that the consensus sequences are actually responsible for the efficiency of transcription. After the sigma factor has bound itself to the -35 to -10 sequences in the promoter region, it will guide the RNA to its start site, the +1 transcription start site. The sigma factor will form a closed complex, at which point it will also unwind the DNA at that area, which will form into an open complex. And then after the formation of the open complex, initiation can occur. And the RNA polymerase will synthesize about 8 to 9 nucleotides before that outcompetes the sigma factor, causing it to dissociate from the core enzyme. And after that dissociation occurs, elongation will occur, and then the RNA polymerase at that step will continue to form the messenger RNA transcript up until the stop codon, and where termination will occur. In prokaryotes, remember, that's whenever we get into whenever the mRNA becomes a hairpin loop for rho-independent termination. At that step, remember, we have the dinad sequence where the two sequences of the opposite sides are actually complementary to each other, and then we have our poly U tail, which will actually break away from the DNA sequence due to the weak hydrogen bonding. For rho-dependent termination, remember, we have a specific DNA sequence that pauses the RNA polymerase, and then we have this enzyme like rho protein factor that will actually require ATP and cause the messenger RNA itself to dissociate based on enzymatic activity. And that actually covers all of the transcription for our prokaryotes. Stay tuned to the next video, and we'll talk about some similarities between prokaryotic and eukaryotic transcription.